Hi everyone, welcome to Applied Stem Cell webinar. This webinar is on iPSC genome editing, particularly on rapid automated cell line gene editing, or RACE. As you know, iPSC has a great potential for both research and therapeutic applications. So basically, iPSCs can be isolated from any adult somatic cells. For example, a patient or healthy donor. It can be fibroblasts or cord blood or PBMC donor cells, and these cells can be reprogrammed to make into induced pluripotent stem cells or iPS cells. And iPS cells can then be either genetically modified to make mutations or correct the mutations, or it can be differentiated into any cell types in a human body, such as neurons, blood cells, hepatocytes, epithelial cells, muscle cells, and cardiomyocytes. And on the other hand, after gene editing, these cells can also be differentiated into all 3 germ layer cells. And these cells can then be used for many different applications, including tissue engineering, disease modeling, and more specifically for regenerative medicine for cell-based gene therapy. So let's just give you an example how we use iPS cells to model disease. On the left panel is where we start with a healthy iPS cell line. So basically, the donor is a healthy individual, and we can derive iPS cells from this healthy individual. And then through gene editing, we can introduce mutations so that we can generate a disease state iPS cells. And these cells can be differentiated into specific cell types that is relevant to the disease state. And then we can compare the isogenic cell line, basically a healthy cell line versus a disease state cell line. And then we can start the mechanism of the disease state cell line as well as use these cells for potential therapeutic applications. And on the right side, this goes the other direction, which is we start with a disease state iPS cells. So these cells are derived from patients with no mutations, for example. And these cells can be then genetically engineered to correct the disease mutation back to wild type gene. And then these 2 different cell lines, the disease iPS cells and the corrected wild type cell lines, can be differentiated into specific cell types. And again, we can compare different profiles. So one thing I want to talk about in this webinar is our high throughput genome editing automated system. So this is a system that is a proprietary technology platform established at Applied Stem Cell. So we're not only able to manipulate iPS cells and make mutations, but also we can actually manipulate multiple cell lines and hundreds of cell lines at the same time using high throughput equipment and processes. So, for example, we use FACS sorting to sort transfected cells. And then we use clone imaging to spot the positive cells or transfected cells. And then we use automated clone picker to pick positive clones. And then after clone expansion, we do next-gen sequencing to genotype the positive clones. So this automated process workflow not only increase the productivity and the scale of our gene editing process, and we also have achieved very high success rate, which is up to 98%. And this also avoids human errors if they were done by manual method. So here are several advantages of our high throughput automated system compared to conventional manual method. So the most advanced characteristic is fast turnaround time. So we are able to cut the turnaround time by more than half. So we were able to achieve up to 60% faster turnaround time. And as I mentioned already, we have a high success rate, over 98%. And we do it in 96-well plate format for high throughput editing. So we can manipulate many cell lines simultaneously in parallel. And then we also have our control master IPS cell line that we can use as a control line to make sure our systems are working in optimal condition. And we have SOP for this automated high throughput process. So for example, here we have 3 different types of mutation: knockout, point mutation, and knock-in. And we are able to save basically 60% time for knockout and point mutation, and 20 to 30% time for making knock-in mutation. So this just summarizes again the advantage of using applied stem cells technology and applied stem cells pluripotent stem cell lines. So first of all, we have developed several pluripotent proprietary iPS cell lines that are currently used by the National Institute of Standards and Technology, NIST. It's a genome editing consortium. So it's a reference iPS cell line. So this cell line is very well characterized and currently used as iPS cell standard. So we provide this particular cell line, our master cell line, for customer to use as either as a control or as a starting material for making mutations or other manipulation to the cell line. And we also provide isogenic clones once we do gene editing. We don't do just pools. Many other providers, they provide pool cells after transfection, but we provide isogenic clones so that those are stable clones for you to further to do your studies. And again, our timeline for project is about 2 to 3 months compared to other manual method, which takes about 5 to 6 months. And our high throughput modification technology allow us to do in a 96-well plate format. So basically hundreds of lines or modifications in parallel. This is really make the process much faster and high throughput compared to the current manual method, which is single modification at a time. So I'm going to give you a few examples in terms of the types of modifications we have made successfully. So case 1 is a PARC2 knockout allele. So we're able to make a biallelic modification on both chromosomes, both alleles. So one is deletion of 10 base pair, and the second is a 4 base pair insertion. And then the characterization indicates that this particular gene, when it's knocked out, affects dopaminergic neuron, and we can see that the expression of some of the markers in DA neurons have been dramatically decreased or disappeared. And case 2 is a reporter line that we made. We used a astrocyte-specific promoter to drive a reporter gene expression. And we were able to see that not only by genotyping that the reporter gene is inserted in the right place on the chromosome, but also expressed the reported gene in a timely fashion. And this is an example of mutation variants in a familiar protein variant example. So basically, this family contains mutation in a particular gene, and we're able to create these mutation variants using gene editing. So we start from wild type cell line, and then made point mutation to specific mutation state that corresponds to what happened in patients. And then when we did an enzymatic activity assay, we see the difference in enzyme activity compared to the wild type iPS cell line. And this is our actually proprietary technology that is to make not just only small mutations, but also we can do chromosome translocation. So this is a very complex modification. So basically, one piece of DNA fragment on chromosome X transferred to another chromosome, so this we have a patent technology, and that allow us to do these big complex modification, including a gene fusion application and chromosome translocation. And so we also have established SOPs and products for differentiating iPS cells to many different lineage cells. So here are just some examples. We are able to differentiate iPS cells to neuronal lineage cells, including neural stem cells, cortical neurons, dopaminergic neurons, motor neurons, astrocytes, microglia, and RPE, skeletal muscle cells, cardiomyocytes. And we also have products that are differentiated to hepatocytes from iPS cells. And we've also done differentiation of iPS cells to more the blood lineage cells, including hematopoietic progenitor cells, T cells, and NK cells, and epithelial cells such as lung cells and parasites, and the blood-brain barrier modeling cell types. And so with the combination of gene editing and iPS cell differentiation, we are equipped with a full set of genetic tools and differentiation tools to make many disease models. So this is the webinar focusing on iPS cell, particularly on high throughput gene editing and iPS cell differentiation. If you have any questions, please contact us. The phone number and email are listed on this particular slide. But in short, so contact us at info@appliedstemcell.com. Thank you very much for your attention.